Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about glycogenolysis. During the last few videos, we focused on the postprandial state, that is what happens after a meal. And since there is a lot of glucose present in the meal, and there is a lot of ATP that can be made from the glucose. So during that time when insulin is high, we talked about the major metabolic pathways that were synthesis pathways. So we talked about glycogen synthesis, fatty acid synthesis, triglyceride synthesis, cholesterol synthesis, synthesis of amphiphilic lipids, and then we talked about synthesis of the non-essential amino acids, which then go on to make proteins, and protein synthesis was discussed in video 3.7. The one synthesis we did not talk about was the synthesis of nucleotides. Nucleotides are the building blocks of nucleic acids, and that's because this nucleotide synthesis or nucleotide metabolism is not related to energy. And this is very important because all 30 trillion cells in your body, all 30 trillion cells require energy at all times. So we'll talk about nucleotide metabolism. Probably the best place to discuss that would be when we talk about the biochemistry of cell division. Okay, so we'll leave that for a future video. And also remember, there's one more synthesis that does not happen after postprandial, and that is glucose synthesis. So insulin does not stimulate glucose synthesis. So why make glucose when there is already a lot of glucose coming from your diet? Okay, so this will talk about it under starvation. So starting from today's video, we will talk about this metabolic state, early starvation. What happens to our metabolism during early starvation? So this is the state between meals. Probably the best example of this state would be somewhere around, let's say, 2 to 3 a.m. Early in the morning when you're fast asleep, you ate your dinner, let's say around 6 or 7 p.m. And now these few hours have passed by and we enter this early starvation state. Now remember, the key question you always need to ask yourself, irrespective what state we are in, is how does your body maintain blood glucose greater than 40 milligrams per deciliter? And this is very important, especially for the red blood cells as well as the brain that get their energy from glucose. So after a meal or early starvation or late starvation or exercising, whatever state a human will be in, this is always a key question to keep at the back of your mind. How does the body maintain blood glucose to make sure it's greater than 40 milligrams per deciliter? So after a meal, the answer is simple. It's your blood glucose is maintained greater than 40. Okay, greater than 40. Why do we choose this 40? Because this is the amount of glucose, critical amount of glucose that is needed in your bloodstream so that the brain can extract this glucose fast enough to provide it with enough energy for its survival. Otherwise, we'll get into a coma. So after a meal, the glucose from your diet will be used to maintain your blood glucose greater than 40. So what about early starvation, the 2 or 3 a.m. in the morning when you are fast asleep or between meals? what maintains your blood glucose greater than 40? And the answer is liver glycogen breakdown. Okay, the breakdown of liver glycogen by a process that we'll talk about, glycogenolysis. This glycogen is broken down into glucose, and that glucose is sent out into the bloodstream, and this glucose will make the red blood cells happy, and it will make the brain cells happy because they can get their ATP from this glucose. We shall also see that although most of your glycogen is in muscles, skeletal muscles, the skeletal muscles do not contribute to maintaining your blood glucose. The glycogen for muscles is needed for the muscles. This glucose that comes out from muscle glycogen does not come out into the blood because it's needed for the muscles. So the only glucose that comes out into the blood to help the red blood cells in the brain is the glucose that's coming out from the liver. So let's look at the key steps that are involved in glycogen breakdown. Okay, and we'll focus first on the liver, breakdown of glycogen or glycogenolysis in the liver, and then we'll talk a little bit about glycogenolysis in the muscles. Recall that glycogen is a very, very highly branched polysaccharide. It is made up of mostly alpha-1,4 glycosidic linkages. And whenever there is a branch coming out. So whenever there is a branch coming out, you will see an alpha-1,6 glycosidic linkage. So the metabolism of glycogen into glucose clearly would require breaking this alpha-1,4 and this alpha-1,6 present at the branch point. So let's look at the breaking of the alpha-1,4 first. Okay, how do we break this alpha-1,4 glycosidic linkage that joins the glucose units in glycogen? This is carried out by the enzyme glycogen phosphorylase. So this enzyme uses inorganic phosphate to break down the alpha-1,4 found in glycogen, and what you get is glucose 1-phosphate, and the glycogen is reduced by 1 unit. So the product that we get is glucose 1-phosphate and not glucose because we are using inorganic phosphate to break down the alpha-1,4 bond. Now recall that the glycogen molecule will have only one reducing end. All the other ends, this will be a non-reducing end, the other end here, the other branch will have a non-reducing end. So glycogen has only one reducing end, all the other terminals on the branches will be non-reducing ends. So there will be many, many non-reducing ends, but only one reducing end. And you'll see why this is important. Glycogen phosphorylase will cleave the alpha-1,4 glycosidic linkage only from the non-reducing end. And it has to be 4 glucose units away from a branch point. So if you look at this picture here, this enzyme will break down alpha-1,4 here, alpha-1,4 here, alpha-1,4 here, and now we are 1, 2, 3, 4 units away from a branch point, and the enzyme will stop working on this branch. This short branch with 4 units away from the branch point, this is called the limit branch. As we mentioned earlier, the enzyme uses phosphate and not water to break down the glycogen. So that's why we call this phosphorylasis instead of hydrolysis. If it used water, then you will get glucose instead of glucose 1-phosphate. This enzyme, glycogen phosphorase, requires pyridoxal phosphate, a cofactor, a coenzyme that we talked about under the synthesis of amino acids, the transamination reaction that we talked about under amino acid synthesis. Now, a glycogen molecule, when you look at it three-dimensional, because of all these branches going in three-dimensional space, this molecule looks like a ball, like a spherical ball. And the enzyme glycogen phosphorylase, so one enzyme will sit here at the non-reducing end, and there's so many non-reducing ends on the glycogen molecule. So we have many enzymes working simultaneously on the glycogen, breaking down the glycogen terminal, the non-reducing terminal, and giving several glucose 1-phosphate at the same time. So there is a rapid formation of glucose 1-phosphate. And this is important because, as we shall see, the glucose 1-phosphate can be converted to glucose, and this glucose can be sent out into the bloodstream from the liver and help maintain the glucose at greater than 40 milligrams per deciliter. And that's critical for, especially for the brain, to get enough ATP energy from the glucose. We shall talk about regulation of metabolism in a future video, but just a few words. Glycogen phosphorylase is closely regulated, and that is done by allosteric regulation as well as covalent modification, especially phosphorylation. Okay, so glycogen phosphorylase will break down the alpha-1,4, and this will be converted to glucose 1-phosphate. Similarly for this, and then it stops when it reaches the limit branch. So what happens to this limit branch, and what breaks the alpha-1,6? Let's look at the next step. So there's another enzyme that is required that's called the debranching enzyme, and this enzyme, we'll see, breaks down the alpha-1,6 glycosidic bond. How does this happen? This debranching enzyme has two types of catalytic activities. One is a transferase activity, and the other is a hydrolase activity. The transferase activity of the debranching enzyme will take these three glucose units on the limit branch and transfer it to another branch. So these three units are now connected to another branch, and that leaves us with only one glucose joined to the glycogen by an alpha-1,6. In the next step, the hydrolytic activity, what we call the glucosidase activity of this debranching enzyme, will hydrolyze the alpha-1,6. So it will break down this alpha-1,6 using water in this case, not phosphate, water. And that results in the formation of glucose, and the branch point is removed from the glycogen. So this means that most of the glycogen is broken down into glucose 1-phosphate due to the phosphorylase activity, and there is also some glucose that will be formed by the action of the debranching enzyme that will break down the alpha-1,6 using water, and that gives us glucose. In the next step, the glucose 1-phosphate is isomerized to glucose 6-phosphate, and that is carried out by the isomerase phosphoglucomutase. Now glucose 6-phosphate, this intermediate here, is a very important intermediate not only for glycogen metabolism. We saw this intermediate form in glycolysis, we saw this intermediate in the hexose monophosphate shunt, and we will see that this intermediate is also formed in gluconeogenesis. In the next step, the enzyme glucose 6-phosphatase will break down glucose 6-phosphate into glucose. So the hydrolysis of glucose 6-phosphate into glucose will release inorganic phosphate, which can be used by glycogen phosphorylase. Now this enzyme glucose 6-phosphatase is found in the endoplasmic reticulum of the liver. We don't find this enzyme in the muscles. It is found in the ER of liver cells. So therefore we require a transport protein, glucose 6-phosphate translocase, that will bring the glucose 6-phosphate from the cytoplasm into the ER, and then once the glucose 6-phosphate is converted to glucose and PI, then this translocase will take out the glucose and PI from the ER into the cytoplasm. Any defect in this system will cause what we call type 1 glycogen storage disease, and we'll talk about these genetic diseases that affect metabolism in future videos. So that's how during early starvation, when you're fast asleep, 2-3 in the morning, your liver glycogen is being broken down into glucose. So there's a lot of glucose that accumulates in your hepatocytes, a lot of glucose present in the hepatocytes. And then this glucose will now be sent out from the hepatocytes into the bloodstream, and the glucose will be exported out by facilitated diffusion, and this occurs through the transport protein that is present in liver cells, and this is GLUT2. Now GLUT2 has a very high Km for glucose, 20 millimolar, which means it has a low affinity for glucose. High Km means low affinity. Compared to other GLUT transport proteins that we'll talk about in the future, they have much more better affinity for glucose, and their Km is much lower, less than 10 millimolar. So since the intracellular glucose in hepatocytes, now this glucose is coming mostly now at this stage from glycogenolysis, and later we'll also see some of it will also come from gluconeogenesis. So the concentration of glucose inside the hepatocyte can very easily reach and exceed 20 millimolar. So although this has a low affinity, it has a high capacity to send this glucose out without getting saturated. So it doesn't get saturated easily because it has a high Km. So although it's a low affinity, it's a very good capacity to transport all this glucose that is very rich in the cells out into the bloodstream. And during early starvation, this glucose will be used now mostly by your brain and your red blood cells. So that's how we make sure that when we are fast asleep, our brain has enough glucose so that we don't get into a coma and do not wake up the next day. A few words about glycogenolysis in muscles. Now, muscles have much more glycogen, about 500 grams of glycogen, compared to a liver, which is only about 100 grams. There's a little bit of glycogen also found in other tissues, such as heart and brain. Now, muscles need their glycogen for their own purposes. They need, especially during exercise, they need to break down the glycogen to glucose 6-phosphate, and they break it down to glucose 6-phosphate They will break down the glycogen to glucose 6-phosphate, very similar to what happens in the liver. But once we reach glucose 6-phosphate, muscles do not have this enzyme glucose 6-phosphatase. So they do not produce glucose. Instead, the glucose 6-phosphate that is produced will enter glycolysis, what we remember the main street we talked about, and then PDH, and then Krebs cycle and the electron transport chain oxidative phosphorylation to give us ATP. Because muscles glycogen is giving you glucose 6-phosphate, you get an extra ATP. So instead of 30 to 32, you get 31 to 33, depending on the shuttle use, because we are not using one ATP for the first reaction of glycolysis, where glucose is converted to glucose 6-phosphate. So just to summarize, in the liver, glycogen is broken down to glucose, which is exported out of the liver cell. In the muscle, this does not happen because muscles do not have glucose 6-phosphatase. Instead, the glycogen is broken down to glucose 6-phosphate, especially during exercise. And then this glucose 6-phosphate is used to produce energy for the muscle cells. Now, muscle cells can produce lactic acid from the glucose. Okay, we talked about this glycolysis, and if there is insufficient oxygen, then we can get lactic acid. Now later we'll see that lactic acid is a source for gluconeogenesis, which is carried out by the liver and the kidneys, and gluconeogenesis will help maintain blood glucose. So indirectly, we could say that muscles, by producing lactic acid, could indirectly maintain blood glucose. Before we leave, a few words about isoenzymes. It's important to note that a protein, such as an enzyme, may have different forms. These are called isoenzymes. So for example, glycogen phosphorylase, there are 3 types of isoenzymes that we know that exist in humans. And these isoenzymes have different tissue distribution. So the symptoms that are related to due to a deficiency of an enzyme really depends on which isoenzyme is affected. Okay, so we'll talk about genetic diseases involving proteins, including enzymes, in future videos. So we won't talk about it now, we'll leave this for future videos. Okay, so during early starvation, the liver glycogen is providing the glucose which will be used by your red blood cells and brain. What about the other cells? How are they going to get their, if they are not going to use glucose, what is going to give them their energy? And the answer is fatty acid breakdown. So the liver glycogenolysis has solved our problem of maintaining glucose greater than 40, and that is important, especially for the brain to get its ATP, and also for the red blood cells to get its ATP. So now what about the other cells? We'll talk about this in the next video, and the other cells will get their ATP from fatty acids. In the next video, we shall look at fatty acid breakdown. Thank you for watching. Once again, we wish you a very happy and a very healthy day. Hopefully, we'll see you soon in the next video.